Welcome to this video on phytochemicals. We will explore the integration of herbal medicine into healthcare and the science behind plant-derived compounds. We will summarize three main areas: the regulatory framework for herbal medicines, the phytochemical origins, and the clinical efficacy of approved formulations. Artemisia annua L is a key example. Let's begin with the macro-regulatory funnel and the integration of herbal medicine into national healthcare systems. Traditional medicine is a significant global healthcare component, driving substantial economic revenue. Key countries show multi-billion-dollar markets. The WHO framework emphasizes integrating it into national systems for patient safety, quality, and access. The Republic Act 8423, or TAMA, formally integrates indigenous medical knowledge into the national healthcare system, establishing a timeline from 1977 to 1997. This legal mandate acknowledges indigenous property rights and ensures fair financial returns. Modern pharmacology heavily relies on botanical origins. Medicines like aspirin, oseltamivir, and artemether are derived from plant sources, demonstrating the close link between natural and synthetic compounds. Regulatory agencies classify botanical products into three tiers based on required evidence: herbal/food supplements, traditionally used herbal products, and herbal medicines. Each tier has distinct requirements for registration and therapeutic claims. Product packaging legally reflects its regulatory tier. A traditionally used product, like ginger, has a THPR number and folkloric use claims. A regulated herbal medicine, like Lagundi, has an HMR number and authorized treatment indications. Scientists often seek a single compound for therapeutic effects. However, plant extracts contain multiple compounds that can demonstrate synergy and be utilized in a multi-targeted approach. This also influences conventional drug development. Synergy and a multi-targeted approach involve a combination of substances yielding a greater effect than individual contributions. This can lead to increased therapeutic effects and a reduced profile of side effects. Plant extracts leverage synergistic and multi-targeted mechanisms. The Artemisia synergy model shows how flavonoids enhance artemisinins' effect. The Salix alba model demonstrates how whole extracts reduce GI side effects compared to single compound aspirin. Whole plant extracts often show superior clinical efficacy. In Artemisia annua, flavonoids enhance artemisinins' action. In Salix alba, salicin combined with other compounds offers selective COX-2 inhibition without GI toxicity. Combination is greater than individual contributions. Whole plant extracts utilize multiple phytochemical classes simultaneously. The multi-targeted synergy of Artemisia annua achieves effectiveness with a 50 mg dose, significantly lower than the 3,000 mg isolated dose. This helps reduce resistance. The pharmacology of synergy in Artemisia annua demonstrates that secondary compounds like flavonoids inhibit multi-drug resistance efflux pumps. This synergistic blockade allows the primary active principle, artemisinin, to accumulate and achieve clinical efficacy. In salicylates and cyclooxygenase inhibition, salicin from Salix alba is transformed into salicylic acid in the liver, providing targeted COX-2 inhibition. This extract offers a superior gastrointestinal safety profile, likely due to synergistic protective botanical tannins. Artemisia annua leaf tea has been used to treat malaria for thousands of years. The active compound artemisinin has its activity enhanced by flavonoids, which themselves lack antimalarial activity. This also decreases resistance to Plasmodium spp. By inhibiting efflux pumps. Salix alba, or white willow, has been studied in clinical trials for osteoarthritis. Its active ingredient, salicin, is a salicylic alcohol derivative with anti-inflammatory and analgesic effects. Flavonoids and tannins may contribute to a synergistic effect, as GI side effects common with aspirin were not observed. We now move to the second section of this video, the biochemical origins of phytochemicals. Plant secondary metabolism branches from primary energy cycles to produce pharmacologically active phytochemicals. Phytochemicals are naturally occurring secondary metabolites used by plants for defense and adaptation, which provide therapeutic effects in human medicine. Three distinct metabolic pathways generate the majority of medicinal plant compounds: the shikimate, malonic acid, and mevalonic acid DOX pathways. These pathways produce various phytochemicals, including alkaloids, lignans, polyketides, and flavonoids. This diagram illustrates the interconnectedness of primary and secondary plant metabolism, focusing on the precursors and initial steps that lead to phytochemical synthesis. The shikimate pathway is a key route for synthesizing aromatic compounds, starting from phosphenylpyruvate and ERYTHROSE4-phosphate. It leads to aromatic amino acids and subsequently to important phytochemical classes like alkaloids and lignins. The shikimate pathway involves a series of enzymatic reactions leading to chorismate, a critical intermediate for synthesizing aromatic amino acids and many other secondary metabolites. The malonic acid pathway uses acetyl-CoA and malonyl-CoA as starting materials. This pathway is crucial for synthesizing polyketides and flavonoids, demonstrating how primary metabolites are diverted to produce complex secondary structures. The synthesis of fatty acids and polyketides highlights the iterative process of condensation and reduction characteristic of the malonic acid pathway. The mevalonate and deoxyzellulose pathways are responsible for producing isoprene units, which are precursors for terpenes and steroids. The two pathways occur in different cellular compartments, demonstrating metabolic compartmentalization in plants. This detailed diagram shows the convergence of the two isoprenoid pathways, the mevalonate pathway in the cytosol, and the DOC/MEP pathway in the plastids, leading to the formation of isopentenyl pyrophosphate, or IPP. The mevalonate pathway in the cytosol utilizes acetyl-CoA to form IPP, which then contributes to various sesquiterpenoids and triterpenoids, including steroids. The DOX/MEP pathway, localized in the plastids, uses pyruvate and glyceraldehyde 3-phosphate to generate IPP, essential for monoterpenoids and diterpenoids. Finally, we will discuss the clinical efficacy and regulatory status of approved herbal medicine formulations. This section summarizes the 10 major herbal medicines recognized and approved under the National Integrated Research Program on Medicinal Plants, or NIRPROMP. These 10 herbal medicines are approved for various indications, including cough, diuresis, anti-gout, anti-diarrhea, and anti-scabies. They are regulated herbal medicines with established clinical trial data. We will now focus on the products developed under NIRPROMP, starting with a few examples. Lagundi, or Vitex Negundo L, is indicated for relief of cough due to common colds, flu, mild to moderate acute bronchitis, and reversible bronchospasm in those over 2 years old. It is available in tablet and syrup formulations. Lagundi technology transfer was completed in 1994 for the tablet and 1999 for the syrup. It has been licensed to several companies, including Pascual Laboratories for ASCOF and Ritem for stop cough. Clinical trials of Lagundi showed decreased cough frequency by the 3rd day compared to placebo, increased airflow, and easier expectoration. Its properties are linked to iridoids, which possess analgesic and anti-inflammatory activities. This image displays a number of Lagundi syrup and tablet products, showcasing the commercialization of this herbal medicine. Here we see additional packaging and commercial products of Lagundi, illustrating the various brand formulations available in the market. The image provides examples of other marketed Lagundi products, which are distributed by different licensed companies. Further packaging examples of Lagundi products demonstrate the diverse commercial presentations of this herbal cough remedy. This visual summary concludes the examples of the licensed Lagundi formulations. Next is Sambong, or Blumea balsamifera L, which is formulated as 250 milligrams and 500 milligrams tablets. It is indicated as a diuretic for mild to moderate congestive heart failure and as an anti-urolithiasis agent for urinary tract stones. The technology transfer document for Sambong was completed in 1988. It is licensed to several companies, including Pascual Laboratories for relief. Sambong is comparable to thiazide diuretics in increasing urinary volume, but, unlike thiazide, does not cause urinary potassium loss. Clinical trials showed higher rates of stone passage compared to placebo or hydration alone. This image displays the commercially available tablet formulation of Sambong, which is used for its diuretic and anti-urolithiasis properties.